వెల్కమ్ బ్యాక్ టు మై ఛానల్ కాంపిటేటివ్ బాటనీ. ఈ వీడియోలో మీరు ఇంట్రొడక్షన్ టు అప్లైడ్ బయాలజీ సెకండ్ పేపర్ అయినటువంటి మేజర్ 2 పేపర్ అయినటువంటి ఇంట్రొడక్షన్ టు అప్లైడ్ బయాలజీ నుంచి థర్డ్ యూనిట్ ఎసెన్షియల్స్ ఆఫ్ బయోటెక్నాలజీ నుంచి 8th టాపిక్ క్లోనింగ్ వెక్టర్స్ అండ్ జెనెటిక్ ఇంజనీరింగ్ అనే టాపిక్ మీద ఆబ్జెక్టివ్ టైప్ ఆఫ్ క్వశ్చన్స్ ఈ వీడియోలో చూడబోతున్నారు. ఈ వీడియో మేజర్ బయాలజీ స్ట్రీమ్ తీసుకున్న స్టూడెంట్స్ అందరికీ కూడా చాలా బాగా యూజ్ అవుతుంది. అంటే B.Sc ఆనర్స్ బయాలజీ ఏ సబ్జెక్ట్ తీసుకున్నా ఈ వీడియో మీకు చాలా బాగా ఉపయోగపడుతుంది. ఎందుకంటే మీరు రాయబోతున్న ఎగ్జామ్ ఆబ్జెక్టివ్ మోడ్లో ఉండబోతుంది కాబట్టి. సో లెట్స్ సీ ద క్వశ్చన్స్ వన్ బై వన్. సో వాట్ ఈజ్ మెంట్ బై జెనెటిక్ ఇంజనీరింగ్? ఆప్షన్స్ ఆర్ selective breeding of organisms, modification of an organism, DNA using biotechnology, cross breeding of plants and animals. So the correct answer is second one, that is modification of an organism's DNA using biotechnology is nothing but genetic engineering. Okay, let's see the second question. What is the purpose of polymerase chain reaction in genetic engineering? So options are cloning genes, amplifying DNA, inserting genes into plasmids. So PCR is nothing but that amplifies the DNA. You can get many copies with PCR technique. So let's see the next question. Which of the following is an application of genetic engineering in agriculture? So options are gene therapy, golden rice with increased vitamin A content, DNA fingerprinting. ఓకే, సో జెనెటిక్ ఇంజనీరింగ్ ప్రోడక్ట్ ఏంటి అంటే అగ్రికల్చర్లో గోల్డెన్ రైస్ విత్ ఇంక్రీజ్డ్ విటమిన్ ఏ విచ్ ఈజ్ ఏ జెనెటిక్ ఇంజనీరింగ్ అప్లికేషన్ ఇన్ అగ్రికల్చర్. సో వాట్ ఈస్ ద ఫంక్షన్ ఆఫ్ CRISPR CIS9 in genetic engineering? Options are DNA amplification, gene editing, protein synthesis. So the correct answer is gene editing is the correct answer. Let's see the next question. What is a transgenic organism in the context of genetic engineering? Options are an organism with genes from another species introduced into its genome, an organism with naturally occurring genetic variations, an organism created through selective breeding. So the correct answer is first one, that is an organism with genes from another species introduced into its genome. Okay, so let's see the next question. Which technique is used to introduce a recombinant plasmid into bacterial cells during cloning? Options are electrophoresis, transformation, southern blotting. So the correct answer is transformation is the correct answer. Let's see the next question. In the context of cloning, define a vector. Options are a type of restriction enzyme, a carrier molecule for DNA fragments, a gene expression enhancer. So the correct answer is a carrier molecule for DNA fragments. Okay, so let's see the next question. Define a vector in the context of genetic engineering. Options are a disease-causing agent, a carrier molecule used to transfer foreign DNA into a host organism, a genetic disorder. So the correct answer is a carrier molecule that used to transfer foreign DNA into a host organism. Let's see the next question. Name the biological method that involves the use of viral vectors for gene delivery. Options are agrobacterium mediated transformation, electroporation, viral transduction. So viral transduction is the correct answer. Let's see the next question. Who proposed the recombinant DNA technology? Options are Weyer Arbor, Stanley Cohen, Herbert Boyer, David Wilson. So the correct answer is second one, Stanley Cohen and Herbert Boyer. They combinedly proposed the recombinant DNA technology for the first time. So let's see the next question. These questions are belonging to fill in the blank type. Okay, you have to fill the blank with appropriate answer. Name a commonly used cloning vector in genetic engineering. So you have to fill the blank. So generally plasmids are the commonly used vectors. Okay, సో ఆన్సర్ కోసం టెన్షన్ అవ్వద్దు, లాస్ట్లో మీకు స్క్రీన్ షేర్ చేస్తాను కాబట్టి అప్పుడు మీరు స్క్రీన్ షాట్ తీసుకోవచ్చు. Recombinant DNA technology allows the creation of dash by combining DNA from different organisms. So, it allows the creation of genetically modified organisms by combining DNA from different organisms. That is a, so the blank is genetically modified organisms. You have to fill this answer. Okay, so the polymerase chain reaction is a technique used to amplify or make multiple copies of a specific dash in a laboratory setting. Yes, PCR that multiplies DNA. Uh, DNA sequences or gene. Okay, you can get multiple copies of gene. Okay, by a process PCR, polymerase chain reaction. So let's see the next question. Genetic engineering involves the manipulation of an organism dash to achieve specific outcomes. So genetic engineering that involves the manipulation of an organism genetic material or DNA. So that is the correct answer. So let's see the last question. Cloning involves the creation of identical copies of a dash, typically a gene or an entire organisms. So the correct answer is you can get identical copies of a DNA fragment. Okay, so DNA fragment is the correct answer. So next, coming to the next category of questions, they are belonging to very short answer type. Let's see the one by one. So what are cloning vectors? Okay, you have to define the vector. A cloning vector is a small piece of DNA into which a piece of foreign DNA is inserted for cloning. Okay, so generally plasmids are generally using vectors present in DNA. They are extranuclear genome. So okay, so let's see the next question. Why is cloning an essential technique in genetic engineering and what application does it have? Yes, cloning enables the creation of identical copies of DNA fragments or organisms, serving applications such as production of specific genes, generally genetically modified organisms, and the study of gene function and regulation. So this is the answer. Okay, let's see the next category question, that is belonging to matching type of question. So plasmids, you have to match the list 1 with list 2. So I'm going to match the list 1 with list 2. So let's see the matching. Plasmids, as I told you before, they are present in bacteria. Extranuclear DNA of bacteria is called plasmids. Bacteriophages, yes, bacteriophages are nothing but they are the viruses. They attack bacteria. They can be used as viral vectors. Cosmids, so cosmids are the lambda phase cos sequences. Bax, bacterial artificial chromosomes are called as bags. Okay, so the correct answer is C. C is the correct answer. So let's see the last category of questions that are belonging to true or false category. Cloning involves the generation of genetically identified identical copies of DNA or organisms. Yes, this statement is true. Okay, so let's see the next question. Cloning vector has the ability to self-replicate and integrate in the host cell. Yes, this is also true. Okay, so these are the questions I have prepared from cloning and genetic engineering. So now I am going to share the screen with answers. You can take screenshots. సో ఈ వీడియో మీకు తప్పకుండా యూజ్ అవుతుంది కదా. ఈ వీడియో మీకు నచ్చిందని కూడా నేను అనుకుంటున్నాను. ఒకవేళ నచ్చితే లైక్ చేయండి, మీ ఫ్రెండ్స్తో షేర్ చేయండి, సబ్స్క్రైబ్ చేయండి. థ్యాంక్ యూ.